So let's talk a little bit about how the actioning of the enzyme, functioning of the enzyme actually occurs. So as already mentioned, that one of the very starting thing as far as the functioning of the enzyme is concerned, a complex has to be formed between the enzyme and the reactant or the substrate, that is known as the enzyme substrate complex. One of the feature of this enzyme substrate complex is that because of the formation of this complex, the activation energy of the reaction is lowered. What is meant by activation energy? It's the minimum amount of energy which is required to carry out a chemical reaction, biochemical reaction. So when that minimum amount of energy required is getting lowered, it is lowered because of the involvement of the enzyme. Of course, then it's much easier, much convenient, or time, it takes much less time for the reaction to get completed. And also, when enzymes or substrates are bind together, it also brought about the very structural parts of those substrates and enzymes, or more than two substrates, in a way aligned. They are aligned in a way that they can bind with each other. Chemical reaction or chemical bonding can take place more easily. So this alignment is also very important. As far as the alignment of the different substrates is also important, because chemical reaction have to depend upon the chemical bonding, which cannot occur if the alignment is not correct. And you can always also keep in mind, whenever you are talking about chemical reaction or anything, when you are studying in science or biology specifically, you have to think in your mind, you have to figure out in your mind the whole picture. And you can, for instance, you can think how what will be the structure of the enzyme, how it will be binding with the substrate. So as a result of this substrate, how let's say there are two or three substrates which have to interact with each other, it will be very important if they are juxtaposed, they are aligned in a way that they can really interact with each other more conveniently and more effectively. One example here we can give, a very important example, you will definitely go through it when you will be studying glycolysis, is the role of hexokinase, one of the very first enzyme which involved in the glycolysis. It leads to the phosphorylation of glucose, giving rise to a glucose 6 phosphate. So again, one molecule is the glucose, the other molecule or other substance the ATP. So ATP has to deliver its phosphate to the glucose to make glucose 6 phosphate. So the binding of the enzyme should with these two substrates should be like that it will align these two substrates so that phosphate can easily be delivered to the glucose in order to form what we say the glucose 6 phosphate. Then also very important aspect of enzymes that they have active sites, the sites with which the substrate will bind. They provide an environment in which the binding between substrates is more stable, is more effective. And of course, that environment it depends upon the very structure, very composition of the active site of the enzymes. The active site can be polar, it can be non-polar, the substrate can be polar, it can be non-polar, it can be hydrophobic, and so on and so forth. So this the providing or facilitate the reaction by providing the most proper suitable environment is also very important aspect of the enzyme substrate reaction. For instance, we can give an example of the pepsin. Pepsin is an enzyme which tends to digest your proteins, specifically in your stomach, and it becomes active when there is more acidity in the stomach. So first the environment have to be acidic, and that acidic environment will lead to the activity of the pepsin, which will ultimately lead to the proper functioning of the pepsin. So in this way, the optimal environment gives rise to the proper functioning or better functioning of the enzyme. And again, it is because this proper functioning is because of the very structure, very composition of the enzyme. Then actually the chemical bonds have to be formed. When we say contorting the chemical bonds, means the substances should be, the substrate should be aligned in a way the chemical bond can be broken or can be formed, as the case may be. Sometimes you have a reaction in which you have to break the chemical bonds. Sometimes, for example, hydrolysis of ATP, and sometimes you have a reaction in which you have to make the chemical bonds. For example, formation of ATP from ATP. So the enzymes in this way make it possible that the chemical bonds are formed or broken in the best possible way. And of course, they do it one of the most effective ways, that they lower the, as I have already told you, the activation energy can be lowered. Here you can see here that when there is no enzyme, the activation energy goes very high, it's very high, and then it has to, I mean, it has to become, it has to, somehow you have to devise a way that this can be lowered, and that way is that you add the enzyme. So when you add this reddish line, or purple line rather, it shows that when you have enzyme, the activation energy is lowered, and you can, this difference shows that how much activation energy is lowered. So that this shows that how, by lowering the activation energy, the enzymes make the reaction to occur more successfully and more effectively. One example again from the metabolism you can take of the Krebs cycle, a tricarboxylic acid cycle. Again, you will, because it's a hub of the cycles, one of the major metabolic cycle is the, or metabolic pathway is this one, the Krebs cycle. You will go through it, and then when you convert citrate to isocitrate, so the molecules of the molecule of the citrate have to be aligned in a way that if a transition form is formed that is known as cis-aconitate, and the enzyme which is involved in cis-aconitase, and that cis-aconitate can then very easily be converted into the citrate. So it can very easily be converted into isocitrate. So this enzyme enzyme substrate transition formation, the formation of the transition product is very important, and that cannot be done if enzyme cannot align the molecules in a way that that transition product can be formed. So how enzyme tend to lower the activation energy? That could be one question. So one of the ways that, because again, whatever enzyme is doing is because of the amino acid it contains, and these amino acids can provide certain ions or chemical groups which tends to make the bonds, which leads to the lowering of the activation energy. Some of the most important bonds, of course, are the covalent bonds. Then once the enzyme substrate product is formed and energy of activation is lowered and reaction can be completed, then after the completion of the reaction, the the substrate enzyme complex is separated or broken down, and the substrate or the product is released, and enzyme come into its native original state. And it's a very important feature of the enzyme action, and you know it, that one of the major feature of enzymes is that they of course tend to make a product with the enzyme, but that is a very transitory product. It's in a transitional stage, and then the enzyme come into its original state without getting changed, and the product is released. So that this is how enzyme work. They lower the activation energy, they align the molecules, but they themselves don't get changed.